Hello.
Hi, folks. Yeah, so it's just us today because Nadira is in Singapore. And we were actually talking about, so we're going to be talking about some terrifying brain diseases, or in particular, one disease, which is Huntington's disease. Um, but yeah, so Ruby was actually talking about, um, she's been working on this. So she's doing a class lecture on Huntington's. And so I think so. One of the things you were saying is that it's easy for you to come up with quiz questions because they're like coming up with trivia questions.
Yeah, I was saying that like during the okay, so backstory is during the pandemic, I started trivia basically. I just like
Mhm.
made trivia for all my friends, and I was just making a lot of multiple choice questions about like random things I was reading on the internet and like interesting facts. And it turns out it like really trained me well to make quiz questions for actual students in class because you have to like think about like what the material is, like what kind of questions you can ask without them like actually looking through the books. And like it needs to be fun and not super obvious. So it really feels like making trivia questions. And like you can gauge like how hard, and like I'll try to make like three hard questions, three easy-ish questions, and then like four like in the middle. And I was just sat there and I was making these questions and I was just like, holy crap, did I actually like pseudo train for this during the pandemic?
I mean, and to me, like that makes sense actually, because I was gonna say the interesting thing about trivia is like you get very rapid feedback on whether the questions were good or not. Like,
Yeah. That's exactly it. And then you like, you get your Google sheet back and you're like looking at the percentages like, oh, that was a good question. That wasn't a good
mm,
question. And because like I've gone through that with so many iterations, like I started realizing what's a good question and what isn't a good question. And so you can kind of like premonition like the results
mhm,
on these
exactly. Yeah,
quizzes.
like it's like an implicit learning thing where you kind of learn what makes a good trivia question. Um,
Yeah.
Also, was your trivia, was that the one where people always wanted one of the categories to be rocks, or was that the other trivia?
I think that's a different trivia, but
Okay,
we
I
could
think
do a
that was
rock
one that my
episode.
neuroscience program did. Yeah, we we I forget where it started, but kind of I think because one of the people was a geologist, and so they kept requesting like, can one of the categories be rocks? And then eventually, like we did do one where just the category was rocks, so like there was pictures of different rocks and like facts about rocks, and it was like pretty bad. But
I had cats. People really liked the cat episode, or like not cat episode, the cat category. And we just like
yeah,
had like breeds of cats. And it was just
oh
like the
right.
photo round, the picture round was just like different kinds of cats. And
Mhm.
people loved that. That was like so much fun. I
Yeah, no,
learned
I
so much about
remember that.
cats.
So like
Yeah.
the yeah, that was the one where like we got the thing and we had to like identify all the different kinds of cats.
Yep. Um anyway, it's now 9 o'clock. Welcome to Philosophizing. We I'm Ruby.
And I'm Nathan.
And
And
today
Nadira
we're gonna
is not here.
talk
Yeah.
and Nadira's in Singapore. So we thought we'd do something slightly different and also to practice this lecture that is on a topic that I am not an expert on. And I realized that this is like literally the format of our stream, except now there'll be slides because I prepared them for a class I'm giving. And I wanted to practice
I
and
feel like
um yeah.
I feel like we've done slides before. Like we did, I think sometime when we did like
Oh,
weird
Halloween.
animals or like weird
Yeah.
bugs or something. Yeah. Anyway, but yeah, cool. Okay. And like, so I put the title as terrifying brain diseases because that's what you're going to
So
talk about.
we're
Right?
just gonna talk
It's like,
one terrifying disease.
yeah, it is making it clickbaity. Like, yes,
Yeah, I you're very
terrifying
good
brain
at making it
diseases.
clickbaity though, I have to say.
Thanks.
Let me
Um,
see if I can. How do I share the screen?
Um, it's the button that's like next to your camera button on Restream.
I actually only have settings, and mute. Ooh, that's not good.
Your settings and mute. You don't have a camera button?
I have a camera button, but just like disable my camera or bring it back, which I just did.
Oh, but then what's next to what's on the right side of the camera button?
Settings.
Oh, it might be just like that your web browser doesn't support that feature then.
Oh no. How did we do it before? Maybe I had to have permissions in the past?
Different browser? Or yeah, maybe permissions.
Yeah. Okay, well, that's a new problem.
While we deal with technical difficulties.
Sorry about that.
I mean, so you could also send me things like if it's online and I could pull it up.
Yes, I can just send this to you. Let's see. I will pull up email. I am emailing you a file. It possibly would be too big because it has videos and stuff, but let's try.
Is it going? Are you sending it to my Gmail or?
I'm gonna send it to.
Because like that has very limited space is available.
Okay, how about I send it to your work?
Okay, sure.
Okay, it's literally just called HD for Huntington's disease.
Oh, I see. Yeah.
Okay, so context of this lecture is, yeah, so I'm part of, like, I'm TAing this class called neurodegenerative diseases, and one of the sections doesn't have a speaker this year, and so the professor was like, "Do you want to give this lecture?" And I was like, "Sure, I need practice giving lectures." I did not think about how much work it was. It took a whole week to make this. So I was like, oh, okay, you know what? Instead of like the 14 kids in this class seeing it, let's just put it on the interwebs as I practice giving it. I actually got to delve into like a lot of the history and stuff. So like I knew kind of peripherally what Huntington's disease was before I like made a whole ass lecture about it, but like I never like delved into the history and like the social aspects of it and like the discovery of the genes. Like I never really thought that deeply about it. It was just like, oh, here's a gene, this is like what it does, it's horrible, like hope you don't have it. Like that's kind of what I learned when I was in undergrad. And revisiting it as a topic as I guess like an adult, I was kind of an adult in college, like it made me appreciate it a lot more. And it was really fun to read some of the historical papers too, which is why I wanted I thought it would be really good.
Yeah, you wanted to go through some
stream topic.
history and yeah.
I always oh my gosh, the share screen thing is back.
Oh, okay, cool.
I found it.
I have not gotten this email yet, so let's just use that. Okay.
Okay. Let's do the thing where.
Wait, so did it just mysteriously reappear?
Yeah, it mysteriously reappeared. I have no idea how.
Weird.
Okay, entire screen. Let's do I guess entire screen. Also share screen. Yeah. Oh, so bizarre.
Okay.
Yeah.
Cool. Okay.
That's a little wackadoodle.
Yes, I can see polyglutamine expansions and Huntington's disease. Wow,
Yeah,
this is like the opposite of a clickbait title.
Well, I'm not. I feel like when you're in a professional setting, you do the opposite of clickbait. And then when you do like real life things, you go I was just trying to like get your face on the screen so I can see feedback, but I think I'm just gonna see like this little Nathan in the corner of my screen, and I'll just have to live with that. Okay. Okay, cool. Yeah, so that's the title of the talk, I guess, lecture. Polyglutamine refers to the CAG, like the CAG expansions that is a part of Huntington's disease. But we'll go into it. So like here's an outline, which I don't think you need. So I'm just going to talk first about how the model, that Huntington's disease is a great model for neurodegenerative diseases, and how the advantages of studying a monogenetic disease, and how like knowing that when like one single gene is involved, it's really good for studying or making investigations, because like at every branch point of the investigation, if it's not related to the gene, you can just like ignore it. So
Huh, uh-huh.
really nice to have like one gene, like one disease. I'm going to tell you a little bit about the history of studying Huntington's disease, and then I'll walk you through like the steps of discovery and and then like finish it with like what we think are possible mechanisms of pathology and how this disease could be like mediated, and like modern strategies for modifying the disease is what I called that section.
Cool. Yeah.
So I'm gonna start really at the beginning. Like this is like the intro that everyone gets, which is like its prevalence is like 1 in 10,000 people, mostly. So that percentage is for European descents. So it's like 1 in
Mhm.
10,000 for people of European descent. It's a little bit lower in people from Asia and Africa. Um, and it presents you like the typical Huntington's disease presents in your late middle age, so like 35, 45, and it's basically begins with personality changes and depression, and then motor onset of symptoms, and then basically you die in like the next 20 years.
Well,
It's
so
fairly horrible.
that's
Go ahead.
surprising. Like I think, I mean, okay, maybe that's kind of interesting that personality changes is like one of the like that seems like a very atypical. I mean, maybe just because I'm used to thinking about like MCI and dementia and that sort of thing, but that seems like a little. It's an interesting like first set of symptoms to have, right?
Yeah, like even sometimes before the motor, the classic chorea part
Yeah.
of the disease, people like people that are close to the individuals that are suffering from Huntington's disease will notice personality changes, and
Mhm.
they'll be affected. And
They'll
like for a
just be
long
like,
time, yeah, I was gonna say for a long time, people once they were diagnosed with it, and they're like, "Oh, they're depressed because of the diagnosis," but also they were actually depressed. Like they're actually just genuinely depressed, not just because they got a fatal disease diagnosis.
was it
Like,
like it's like depression is actually part of the process of the
yeah,
disease. Yeah.
yeah, yeah.
Um, I think do they so like the personality changes are just kind of like people like around them notice that they are like kind of like what kind of personality changes and like what areas of the brain are affected first?
Yeah, so, like, I think it varies with different people, but
Mhm.
usually they say, like, executive function, which kind of is, like, decision
Mm,
making
mhm,
and, like, do like, what do you do first? Like, what is important?
mhm,
And also, like, holding in, like, rage issues and things like that. Like,
mm,
um,
mhm,
there's, like, a whole section I'm gonna talk a little bit about, like, how, like, the propaganda and the whole eugenics movement, like, had like, went really horribly wrong. But there genuinely are cognitive deficits, and that's like a major pillar, like landmark phenotype of this disease, as later on you actually do develop dementia, not the same way as Alzheimer's dementia happens, like where you lose memory first. Like this is like the other parts come first.
mhm.
um, so here are two famous, well, one is a real person. So on the left is Woody Guthrie, who is a famous folk singer. I had no idea who this person was until I looked him up, because like this is just like the intro slide of the dude. And this guy wrote "This Land Is Your Land." Like, you know that song you sing like in
Mhm.
school? This land is
Yeah, the,
your
like,
land.
weirdly nationalistic one. Or, like,
Yeah.
the weirdly colonial one. Yeah.
Yeah, that song. Yeah, so he wrote the song. He was like a major folk singer in the 40s, and he suffered from Huntington's disease. And the important thing, well, important thing, there's important things about his career and stuff, but how he relates to Huntington's disease is because of how prominent he was as a person. So like I feel like every major neural disease has like a poster person of it, and so he's the poster person for this disease. And his wife actually started a committee to combat Huntington's disease after he died, and that like grew into this like multi-country 38 country international Huntington's disease like association, and like
Oh
it was really
wow.
focused on like the social parts of Huntington's disease, like support groups, like how to deal with it, like family, like how to talk about it, like how to get genetic testing, like the whole like very like the daily like living with it. And it was really concentrated on like helping people who had gotten or had the diagnosis or are living through it, and the people around them that are living with it. So it's like a major organization, is still going today, and I'm in fact I'm possibly might be showing a video if it lets me in a bit. And for those who were not born in the era like that had to sing these songs in elementary school like I did, another other famous like fictional characters are possibly this character 13 from shoot, what's this house, it's really written on
Yeah.
the guide. House MD, yeah. So she was one of the main characters who
Why is she
like
named
throughout
13?
I think the reason she's named 13 is because she was like the 13th applicant or something for the job. And it just like stuck. I forgot her real name. I just
Oh,
like
okay. Yeah.
yeah, this
Okay,
actor
so
Olivia
that's
Wilde. Yeah.
okay. Yeah. Yeah. Yeah.
Yeah, it's not Olivia Wilde. Olivia Wilde probably does not have Huntington's disease. It's just her character, like her character arc had a
Anyway,
whole
cool.
Huntington's disease
But
arc.
like, is the
Yeah.
character named 13?
Um,
Or
I will look up before I
sorry,
get
this is
dyslexia what her
irrelevant.
real name. No, it's cool. It's cool. I should be like, this is the character Dr. Something instead of like her name is 13.
I just
Before
don't
I was
think.
making
Yeah.
this, I like thought her name was 19 in my brain. I was just like, yeah, Agent 19. I was like, wait, that's not right. I'm mixing
You're like,
my
wait, no,
stories.
that's wrong show. Yeah.
Wrong show. But yeah, she so
Anyway, yeah. You're
yeah, so
getting a
those
preview of the kinds of questions that people in my class will
yes,
ask.
no, I told Nathan to be the class of one and the internet to be the rest of the class to practice this. But yeah, so anyway, so she was like yeah, if you didn't know any like um what's it called, like representations of it. So I thought I would start from the beginning, like 19th century beginning,
Mhm.
with a story. So this is a story I found in a book by Alex Wexler. And so she had like she's a historian, and so she went to the East Hamptons and like dug through archives of newspapers and stuff, and she like found like just tracing the history of Huntington's disease in that community. And she put this in like one of the first chapters in the book, and so I thought I would share this excerpt with it. So it's a short excerpt from the 1806 Suffolk Gazette, and it's a news communication from East Hampton describing the tragic death of a woman named Phoebe Hedges, who walked into the sea and never looked back because, quote, this extraordinary step is attributed to her extreme dread of the disorder called St. Vitus's dance, which she began to be affected with, and for which her mother has had a great degree. And so in the 19th century, it was like this like dancing disease called St. Vitus dance, like was already known about, likely because like in the 1500s in France there was this like whole dancing plague, which is a different Korea
Oh
in its own.
yeah.
But that's
Yeah.
yeah, that like we can talk about that plague in another
Mhm.
episode. But like a disease, like an outbreak of people dancing as like the phenotype was like known of. And so they called it chorea. Chorea is Greek for the word dance, like choreography. So yeah, so these movements, these chorea movements are like a classic motor symptom of Huntington's disease, and it's seen in many patients, or most patients, um except for juvenile. See, I always have these like random like side things,
Mhm.
except for juvenile expressions of juvenile Huntington's disease, where they just like skip straight to the like almost Parkinson like stiff part of it. So anyway,
So the kids don't get the like cognitive, or they don't get
yeah,
the
I think
chorea
they
at the
like
end.
fast forwarded the first part because like the chorea is like like early motor things, and I'll have like a timeline of like typical symptoms, and then I'll show you like the speed of like how people progress if you have like more repeats. Okay, so like the first written mentions of Huntington's disease, or like some of the first mentions of it, was in letters by Charles Oscar Waters, and that was published in like the 1840s. And he described a form of chorea he had called the malgrams, And in the 1960s, it was also described by a neurologist in Norway called Johan Lund, and he called it Setesdøya after a valley in southern Norway. And so even like in the 1800s, they were seeing like pockets of people having this disease.
Huh.
Like,
Uh-huh.
and they would like, it would be like, in this valley in southern Norway, there was like this like, outbreak of people with, this chorea, like sorry, Solastrosia disease. But like these are all like kind of like not broad publications. They were just like written locally. And so Huntington's disease is not named after either of these people. It's neither named after Charles Waters or Johan Lund. It's actually named after George Huntington, and that's probably because he came in. Oops. Ah,
What
I
happened?
should I fast forwarded too
Oh,
many.
I
So
was
that's
gonna.
probably
So can
because. Oh yeah, go ahead, go.
I was gonna ask. So why was it happening kind of in these geographically like why was it clustering geographically?
Well, it's likely because they were related to each other.
Oh, I see. Yeah.
That's like the main
So
one.
that
It's
was
like it's
like where the
hereditary,
signals
right?
were.
Yeah.
Yeah.
And so there would be like pockets of it because they were all like semi-related to each other, and like this whole village would have like one person in common to them. So, and that person was afflicted, and so you would get and it's because this is an autosomal dominant and highly penetrant disease, which I'll talk about soon after I finish this history section, which I think is actually quite fun, right? Like this feels like
Yeah.
a normal
Yeah.
so okay, so coming in with the award for best description, and also probably because he that's why it's named after him. It comes George Huntington, whose father and grandfather practiced medicine in the East Hamptons of, I guess, New York area. And they kept like these logs, like really long like ledgers of like patients they've had, and they like kept track of like people who had chorea. And so he described them, and he also like was training at the time, and so he like got to see many of these patients, and he made these descriptions. So he wrote this essay in his one and only publication in his entire career called On Korea, which is pictured here on the bottom left. And yeah, I'm going to read a little bit from it because I think it's this description is so apt, like it's still good now. So the disease commonly begins by slight twitchings in the muscles of the face, which gradually increases in violence and variety. The eyelids are kept winking, the brows are corrugated and then elevated. The nose is screwed first to one side, then to the other. The mouth is drawn into various directions, giving the patient the most ludicrous appearance imaginable. As the disease
I'm
progresses, the
just really
mind.
imagining like I'm just really imagining like you go to the doctor, they're just like your nose is screwed. That means you have
Yeah,
Huntington's
it's
disease.
screwed to the left and then to the right.
And then it's this like fatal disease.
Yeah. And as the disease progresses, the mind becomes more or less impaired, in many amounting to insanity, while in other minds and body gradually fail until death relieves them of their suffering. When either or both of the parents have shown manifestations of the disease, one or more of the offspring invariably suffers from the condition. It never skips a generation to again manifest in another. Like that is such a good description.
Yeah.
Like I understand
Oh
why it's
yeah,
named
he
after
got
him.
like
And
he got a
what's
lot.
really
Like he got like the pattern of inheritance. Yeah, like
exactly like what's so cool about him was that he noticed how terrible the disease was, and he had the foresight and intuition to like protect his patients too. Like for example, like when William Osler, who's like who also wrote this book in 1894, he like reached out to him and was like, hey, can I interview these patients that you wrote about in your on Korea? He was just like, nah, these are people are living their lives, like leave them alone. Do not talk to us. And like William Osler could not talk to these patients because he didn't know who they are, because they had like confidentiality in the 1800s, which
wow.
like was such good foresight, because there's a whole like movement of eugenics coming up, and he
Mhm.
was just like, nah, like protect these people. They were like normal humans, that living their lives, and they were like well respected people in the community. Like this was the era before like any stigma like towards having this disease, because likely because no one really noticed like that heritability bit until like around this era, because this was like around like the late 1800s was when they were like rediscovering Mendel's like like writings about his like piece, and like people were like really thinking about heritability and like what breeding was happening in
Mhm.
livestock and in like and in plants and stuff. So anyway, like this was like that era.
So there wasn't and there
So
so there wasn't stigma around it because it was very clear, like, okay, this is just like a terrifying thing that can happen to you.
yeah, they were just and also like they had like really low life expectancies. Like people were dying around
Mhm.
40 anyway. And so it was kind of like we don't really know like why, or it could be like the plague again, like the dancing plague of 1500s. Yeah, so generally like the people that suffered from Huntington's like would be well respected members of the society. um and that like that was like the reality, and then there was like the propaganda afterwards during the eugenics movement, which we're going to skip a few years, and like where they like try to like paint that in a really different light. um but anyway, uh not to go into dark, but here's like a video from the society that I told you about. So like the Guthrie's, Marjorie Guthrie's society, the Huntington's Disease Society of America, which is that's the current name of it. They will have like these videos showing patients with disease. Yeah.
We're not really getting the audio, but I can see the subtitles.
Ah,
That's okay. I think it's just
yeah.
not coming
We
through
can
the
we can
stream.
drop the
Yeah.
link. We can drop the link later. But it's basically a video of a patient like who has volunteered their time and to like help with advocating like diagnosing and helping people with Huntington's disease. So you can clearly see he's suffering from the disease. You can see the chorea, you can see like the the things that Huntington's described, like the nose screwing, the mouth positions, like you can see it already. Like it was a very
Mhm.
good description. So characteristic of symptoms. So here is like a graph. Where this middle line is where the chorea and the motor deficits start coming in, and then this is like birth, and this is like advance, and then like death. And so um and the blue line is your functional abilities. It's just like your daily lives, like getting yourself to and from work and like doing normal things. And you can see this like sharp decline that starts happening when you hit 45-ish, year 45-ish, plus or minus. And you can see these symptoms increasing. So this chorea starts coming up, and then it just like really and then same with dementia and then the motor impairment. And so usually these patients actually die of after they've lost motor control of their breathing, um, and like will swallow incorrectly and get an infection in the lungs and things like usually
Mhm.
like typically like dementia, like how people die from dementia is like other reasons. It's just um yeah, and so like this is just like a way of like framing how it starts, and how it's like this is like just by symptoms, and then this is by like how we're trying to frame it now in the modern era. Like we're trying to frame it like you're predisposed to the disease if you have these triplicate repeats, the polyglutamate repeats in the
Mhm.
gene. Like even if you have zero symptoms, like right here you have zero symptoms, like all the way in,
Right.
like basically here.
And so you have this gene that just has this CAG sequence in it, like,
Yeah,
a
it's
whole
just
bunch of times. Yeah.
yep, a whole bunch of times, and like it's just asymptomatic,
Uh-huh.
and then like in the late stages it'll look like this. So here's a
Wow.
coronal section of the brain. So on the left is just like a healthy brain slice. This is what it should look like. And then on the right is what happens when you suffer from Huntington's. And you can see there's like a lot of loss of in the cortex, like just like the white matter and gray matter, like these huge valleys and gaps here. And then you can also like especially see this massive ventricle, like
Right.
that's
It's just
usually
like
like barely visible.
there's just like no striatum. Like
Yeah. And that's actually the thing. Like it like atrophies the like like starting in like the caudate and
Yes.
putamen, like here in the basal ganglia area. So you can see this like ventricle on the
Right.
right. Like
Mhm.
it's basically like all of this like basal ganglia area.
Yeah.
Which
So for folks who aren't neuroanatomists, kind of what we're talking about is so kind of in this photo on the left, this so kind of where this red arrow is pointing. Normally, there's like a very small pocket of fluid there, and then this darker colored tissue is part of the basal ganglia, which is also called the striatum, and it controls like motor movement. And what you can see is in the Huntington's like brain that has basically all died away, and it's kind of been just this fluid filled pocket has expanded into there just because there isn't any tissue in there anymore.
yeah.
Yeah.
And I have this whole diagram about the striatum and how the basal ganglia works. But this is like kind of refresher from like the actual class I'm teaching, because we talked about Parkinson's last week, and so this week it's like the opposite of it. So in Parkinson's, the direct pathway is really affected, and so they end up with basically they're like frozen and stiff. And Huntington's is almost like the opposite version of this, where in fact it's actually the indirect pathway that gets affected most. And I have like a call response question and answer situation. I was like, ugh, during the class, but it just says it's this pathway that matters for this, because by these neurons dying, basically, which usually inhibits the this other part of the brain, the substantial the STN. And then like by reducing that inhibition, you like basically activate this area, and then like it results in the hyperkinetic movements, so the dancing basically. And currently, well, the FDA doesn't have any therapeutics that specifically treat Huntington's. They just have symptomatic treatments. And so here is a entire chart of symptomatic management of Huntington's disease. And if you look kind of down this table, like you start to realize that it's really derived from like anecdotal clinical experience. It's kind of like doctors are like, oh, you're moving too much. Like, let's give you this other drug that like makes people move less. So there's like no disease modifying interventions currently available, like just like on the market. So everything is in clinical trials, and we'll talk some about that if we have time later. And like a lot of them just like alleviate these symptoms. You can see there's like vesicular transport inhibitors, there's like a bunch of drugs for like schizophrenia, there's things for antipsychotics. Like the majority of these are actually antipsychotics. And then it changes, right? Like as your disease progresses to more like stiff Parkinsonian things, they'll start giving you back dopamine. They'll just do all
Right.
sorts of things to try and like preserve your function for the longest amount of time. So anyway, so now we're back. We skipped a couple years, and two world wars and depression and the horrible eugenics situation, and we're gonna skip forward to actual an era of discovery. So this is the space age. So in the 1950s, which space age, this neurologist named Americo Negrete was spending a year in this town in Venezuela called St. Louis, so in St. Louis, and it was near this lake called Lake Maracaibo. I'm just butchering all the Spanish names. But he was there for the national service. And so while he was there, he observed a lot of like children and young people who would like be like look like they were drunk. They would like stagger home and stagger around. He'd be like, "What's going on?" And the villagers are like, "No, those kids are not drunk. They have, the bad disease." And and so he took really good like histories and like documented these patients. And so these are like juvenile Huntington. So they like skipped the first half of the symptoms from like the hyper movement and stuff straight into like the Parkinsonian like stiff like non-movement. And they were like really severe, really young. And so he took videos of this and he wrote it up in Spanish, and like no one read it for 10 years. And until one of his students actually took these videos to a conference called the Centennial Symposium of Huntington's Chorea. And that's when actually people saw the videos and they're like, oh my gosh, like these children have
Like they're
Huntington's
just like
disease.
they're just watching it and they're just like, whoa, this can happen in children.
Yeah. And it was like, oh no, or we're in the room that is like motor. Hold on. All right, there we go. Um, and so it turns out this population of humans with Huntington's and like the village around this lake is actually the largest single population of Huntington's in the world. And it's scattered like throughout the 3-4 villages around this lake. And so there's over like he found over 300 people, and they were like all 100% at least 50% risk of having Huntington's disease. And so after he wrote about this, 10 years later, one of his students brought it to a conference, showed it to people, and some really important people were in the audience. So one of the people in the audience, so Milton Wexler was a like he was a geneticist. His wife died of Huntington's disease, and so his daughter Nancy, who's pictured in the bottom left, was in the audience of like where she saw this video, and she was really important because Milton and Milton started the foundation, the scientific half of the Guthries. So the Guthries went for all the social stuff. The Wexlers went for like the scientific way of like advocacy, like science advocacy. They were like, we need to find a gene, we need to like fix this disease with science. And so Alice Wexler was part of this this movement, and she basically organized 20 years worth of trips to Venezuela. Basically, she went to Venezuela a lot and collected like 4,000 blood samples from like everyone in the town. And she was like known as like the white-haired lady, and like so she documented over 18,000 individuals and established these pedigrees, like the family trees, basically. And they were able to trace the origin of the disease back to a single woman named Maria Concepcion, who lived in the area in the 1800s. It's like she was like family treeing the entire valley in that area in Venezuela. And this is really important because that depth of work really like allowed them to like find ideal families for genetic linkage analysis, and that allowed us to eventually narrow down the location of the Huntington's gene to like a 500 kilobyte segment between like this part and this part of the arm of the 4th chromosome. And here is a picture of one of these pedigrees that she made. So in circles is like, I think circles are women and squares are men, and in black, if it's colored, it's people afflicted with Huntington's. And if you, so the way genetic gene linkage works is that if you treat each person as a meiotic event, and like if you remember like in biology, like meiotic events are like when the chromosomes like line up to each other and then they like cross over and then they swap some genes. And this is like the reason like we have sex and stuff. Anyway, so if you treat each human as a meiotic event and you count enough each one as like a data point, you can like find areas where so the theory behind it is that the gene, if there's genes next to the gene of disease, those genes are more likely to be moved with it when during the meiotic events than genes that are further away. And so if you have enough people, you can find statistically where the gene could possibly be. So like by crossover events, by like crossover recrossover events, like the recombinations, like you're like, oh, then it must be near here. And if you combine that with genes that have high polymorphisms just naturally in the environment, so for example, like if you took my genome and Nathan's genome, there's just gonna be a bunch of genes that are like, I'll have really long versions of it and he'll have really short versions of it. It's just like a natural variation, and it's like non-coding or coding, it doesn't matter, and doesn't have any phenotypes. It's just like variations. If you use those genes as the marker genes, it's really nice because you can pull them out in a gel. And if those are the genes that are near Huntington's, it'll help us like find it basically. And so,
So kind of,
um,
so what you'll do is, so kind of, you'll take people who have Huntington's and don't, and then you'll kind of chop up their, so kind of, you'll take, so, and like, this is just like from my knowledge, so correct me if I'm wrong, but kind of, you can kind of chop their DNA into different segments based on like where different kinds of markers are. And then you can look at the length of the segment and see like kind of, does Huntington's consistently associate with like this one particular chunk? And like in the, like, is it the people who have this really long, where this chunk is really long, also tend to have Huntington's disease?
yeah, that's exactly it. So the
Okay.
chopping up is done by restriction enzymes. So I think like the correct name for restriction enzyme. Oh, actually, I have a call response part here. So, well, maybe I could practice it. Okay, so um, one of the like before I deviate too far from it, one of the
Mhm.
advantages of Huntington's is that it's you have these like really deep pedigrees, so like the patients don't die before they reproduce, so you can have like longevity. So this doesn't work as well. You can't, it's hard to do linkage analysis when there's not enough subjects. It's like so rare, and it's not like you don't have like multiple generations. Like here there's like 4 generations you can follow back. It's also difficult if the disease is not fully penetrant. So penetrant means like if you got the gene, you're gonna show symptoms. Like, so like this is a fully penetrant autosomally dominant gene. So therefore, like it's a really reliable phenotype. Basically, it's like I mean it's gonna like vary in like when it onsets and stuff, but like you will eventually
Yeah,
experience
like, which
Huntington's
is
if
good
you have
because
the
it's
genes.
yeah, because it's harder to do this analysis, I guess, kind of if the clinical presentation isn't that tightly coupled to the gene, right? Like it's hard to do
Yeah,
this analysis if like most of the people who have the gene don't like get the
yeah.
disease, for
Don't get the
example.
disease.
Um,
Yeah,
can I show
exactly.
people kind of like what you were talking about the chunks of DNA? Because I think that
Yeah, yeah, yeah.
is
So the chunk of DNA, this is the third point. The third point is that like there's useful DNA markers, and they're called restriction fragment length polymorphisms, which means if you use these restriction enzymes and just like cut up my DNA and you cut up Nathan's DNA, like the same restriction enzymes, there's going to be certain parts that are longer, bigger in mine, and some longer, bigger in his, and smaller. Do you know what I mean? There'll be differences. Um, is Nathan? Are you showing
yeah.
them
Oh
a?
yeah. Yeah, yeah. I'm showing them some gels. So kind of, yeah, so when we're talking about like looking at the length of the DNA segments, we're talking about something like this. So kind of the idea is that like the restriction enzyme always cuts the DNA at a certain sequence. So it'll cut the DNA at a sequence that's like ACCG or or like CCTG or something. And then and it'll cut, so kind of there's a bunch of that sequence, so it'll make a bunch of different kind of chunks of random size. But what we were talking about is that kind of, because people have different DNA, in one person kind of there will be a chunk that's like 500 base pairs long. There's 500 letters between the two spots that it cut it. And then another person that might be like 700 base pairs long. And so kind of what you can do is you can actually then you cut up the DNA, you basically put it into what's called this agarose gel, which is kind of a filter that filters DNA by size. And that's what's going on here. Is so this is showing small DNA kind of ends up at this part of the filter, and large chunks of DNA end up kind of at this part of the filter. And so you can actually use this to see kind of like what size of chunks does any particular person have. And then you could see like, okay, do people who have Huntington's always tend to have, say, this chunk around 410 base pairs?
Yeah, exactly. And they're spread out like throughout your genome, like these different size chunks. And so
Mhm.
basically they're looking for chunks that are more similar amongst people who have Huntington's. disease. And like if with enough numbers you can like statistically find that. And that's actually how they first like identified like close location. Like they're like, yeah, it's somewhere in between these 500 base pairs, because they'll have a one of these polymorphisms in these areas. And they're like, okay, so somewhere between here and here there's like a lot of it. A lot of patients have that. And so they're like, okay, these are the likely locations for it. And so um that's actually how they the linkage stuff happened. And they also like realized that the actual gene was a different size. And so like the problem with Huntington's, like compared to like other genetic diseases, is like it's not like the actual gene, there's nothing wrong with it. It's not like flipped in, there's no frame shift, like it's actually fine. It's just there's a chunk inside it that is just massively repeated and long. And so that actually was identified as like that kind of repeat was found as like a disease model, a disease gene before. So for example, here on the left, this is just a paper showing what happens in spinal muscular dystrophy. And you can see like this is the diseased version, and they'll have like this many copies of the CAG, and then like this is what it looks like in a normal person or in a control non-spinal muscular atrophied person. And so like this kind of like repeat as the mutation like was recently like found like back then in 1991. They were like, oh yeah, this could be the disease, a way of the disease. And so then came the cloning version of this, and then they found that it was also true in Huntington's. And so here is a drawing of the just like a schematic of what it looks like. You can see it's at like this tip end of the chromosome 4, and it's this is like locus P4 16.3, and it was initially labeled IT15 for interesting transcript 15. And eventually it's named HTT. Yeah, so back in the day, back then when they're like looking for things, they'll just be like interesting gene 1, interesting gene 2, interesting 3. So it was interesting gene 15 at the time.
Okay,
And you notice it's like in this like microsatellite region here, and so that just means it's like vulnerable, and it's like once you have a couple repeats you can like repeat more. And that's pretty much kind of all we know about the actual gene. Like we don't know what exactly it does, but we do know it's important like if you don't have it. So for example, or if you have multiple copies of it. So here's just another schematic. So here's the chromosome 4. Here is like a version where it's normal size, normal length, and then you get like normal healthy neurons. And if you get like too many repeats of it, then you get like sad degenerating neurons.
and so why does that happen?
Why does the multiple
Yeah.
repeats cause
Yeah.
death? That is a great question, and it's an actively being worked on. I'm going to show a schematic later on with like all the
Like, I mean,
hypotheses.
I guess also, why does this only kill neurons and not other
Yeah,
things?
why does it go for those striatal neurons, right? Like in the caudate putamen and the basal ganglia,
Right.
like
I
why
mean,
those first?
and I mean, also, like, why is, I mean, I was even wondering just like, why is this a brain disease? And like, do other tissues not express this protein or?
Yeah, that is such a good question, because this is like the
Nothing.
exact next slide. So the first thing
Okay.
is
Yeah.
it's actually expressed everywhere. So this is
Okay.
single cell sequencing actually that recently dropped in like March. I guess 3 months ago is kind of recent. So this is like this sequencing. So each dot, you can barely see the dots here because they're all clustered. So this is two-dimensional map of all the neurons in the brain of a mouse, and then in basically purple, red, orange is like where Huntington's gene is naturally expressed. And you can see distinctly in these areas there's no Huntington. So like here is some vascular cells, like in like the smooth muscle, like blood vessels that also get pulled out in this kind of analysis. They have no Huntingtons in them, but like literally every brain neuron expresses Huntington. And we're like,
Great.
what does this gene do? And it actually varies. So here is an older study where they just compared like its expression in like different parts of the human brain. And this is like from anatomy, like they took dead patients' brains and like literally measured the amount of Huntingtons in them, and they could see like it was expressed everywhere. It's like not more in one place than in another, which is kind of weird. And it's like, why is it so severely affected? And so here's one possible reason that striatal neurons are most vulnerable. So there's this thing called somatic instability. So in the mice striatum, the CAG, the expansions happens more rapidly in certain cells than others, and so they think so. This is how it works. So when you have cells that replicate, basically, sorry, when you have cells that have like really similar sequences over and over again, basically they can fold up back to back with each other and create these hairpins. And so when you have a hairpin, then it just like kind of like disappears, and then like the the parts that are not hairpinned will just be they will just match back on when oh my gosh, this is not a good way to explain it.
Right.
And it will
So okay,
it
so
will
you
cause a
like so
like
the hairpin kind of sticks out like it's linear, and then the hairpin kind of sticks out.
yeah, so it seems shorter than it actually is because the hairpin
I see.
hides
Yeah.
the length of it, and so then it'll like repeat it. It'll make the correct amount because like there's DNA repair, right? There like there your like every cell has its natural like, oh shoot, we fucked up, let's fix it. This is like their fix it system, but their fix it system is actually making it worse because it doesn't recognize the hairpins. They had no idea the hairpins happened. They're like, oh shoot, it's incorrect on this side. Like let's fix it by adding all these nucleotides. And so like with multiple times, like with these hairpins every over and over again, you can expect these repeats to get longer and longer. And so it's like a like feed forward calamity.
And so
um
this is just a thing that can happen anywhere where you have like the sequence CAG in the
yeah,
DNA.
yeah.
Yeah.
Basically,
Mhm.
a lot of places that have repeats you can end up with hairpins, and like this is like an error that can happen. And so this error is called like a slip stranded structure. And yeah, that's like one of the reasons people think. Another possible reason that it's in the striatum is that the brain derived neurotropic factor is like lowest in the striatum, and so like they're like possible correlation, like possible reasons there.
Right.
um
So maybe the BDNF protects neurons against this toxicity.
mhm. And it's just like not there. Um,
Mhm.
there's a couple more like examples for like possible
I was
toxicity.
I was gonna say though, so going back to the why is it in the striatum? So kind of the
Mm,
hypothesis with that that slipping thing. So the idea is that the striatal cells actually have more repeats in them than the other cells in the body, or
yeah, yeah.
yeah,
That's the idea. Like when you're developing, like during
I see.
development, like this is the
And
thing
is
that
that like,
happens.
I mean, is it known that that happens? Like, do they like that? Seems like something you could test, right?
Yes, yeah. So they did.
Uh-huh.
They test that in mice. So that is true
I
in
see.
mice. I'm not sure if
Mhm.
they tested in humans, but that is true in mice striatum. And so
Cool.
that's why
Okay.
that theory is there. Yeah. There's
So
like
the
other
repeats
theories.
accumulate more in the striatum, and therefore it attacks the striatum more. Okay.
Correct. Yeah. Um, there's also like here are some other theories that have been outlined. So they think it could be from monomer conformation. So just literally the shape of it. So I'm actually gonna walk through like all the different versions of it in a bit. But I remember at the beginning I was like, this is a great disease to study because it tells us a lot about other diseases. And that's because all the polyglutamate diseases, Huntington's is one of them. There's like 9 others. Here's a chart of all other ones, including SMBA, which I talked about earlier, which is that androgen receptor situation that where people were like, oh, expanded poly CAG repeats like could cause diseases. Like all of these are poly CAG problems, like, and they're all
Mhm.
neuro diseases. um so like studying one of these is studying all of them, kind of studying all of them. Like they present differently, they have different like problems, but like they have similar like genetic like problems. um I think that's all I wanted to say with the slide. Oh, and like where it affects the brain is different too, right? So like here's just like all those genes I showed you in the chart before, and I'm just like arrowing them into different parts of the brain that they affect. And you can see like Huntington's really affects the striatum. and cortex and things. But like these SCA ones really affect the cerebellum, and the inferior olive and things like that. What do I want to say about here? Oh yeah, and these like repeats not don't just happen as CAG repeats. So like glutamine diseases, sorry, not probably disease, tandem repeat diseases also occur with different patterns. So for example, a really famous one is Fragile X. This one is a CGG repeat, and that represents another kind of disease that happens on a different part of the chromosome and has also really severe neurodegenerative development errors. And it doesn't, and like the length of the repeats is different for every disease. Like you can have, in Huntington's you require like 40 plus copies of CAGs, like repeats, like the tandem repeats need to be like 40 plus for you to like have disease like presentation. But like it could be very different in Fragile X, in all of these other diseases that I'm showing in this graph.
Well, I think it's different mechanisms, right? Like I used to work in a lab that did some Fragile X stuff, and I think one of the thoughts is actually when that gene picks up repeats, the gene gets expressed less. Kind of the cell kind of can't figure out what to do with it, so it just stops making that protein, and that protein is like important for the brain, so that's not good. Yeah.
But there are like some trends, like if you like squinted, for example, you can kind of see this like downward trend, like here, not downward trend, but like this diagonal trend, and it basically means that it's like inversely correlated, like the length of the repeat, like how many times the tandem repeat it happens, kind of correlates to the onset of the disease. So like when it like hits the patient, which is a continuing pattern you see even within the same disease.
Yeah.
So
which makes sense.
yeah, like for example, like in the Venezuelan population, where they had all these children with juvenile versions of Huntington's disease, they had like some were like homozygous for this alterated version of Huntington's, and many of them had over 50 copies of the repeats. And you can see like it's like earlier onsets, like more massive, you see more severe symptoms earlier on in the disease cycle. Here's just
you said less than 10 years old. Like for
I know.
Yeah.
No, and like some even earlier, like 2 years old. It's like really sad. This is just another graph showing that it also is true inside Huntington's disease. You see this correlation, but also you see this variation too, like within. So like these people have the same length and they have like age of onset, like the difference between like 20 and like in that same row, like 55. That's a big difference. It's like 30 years. There's like variation, right? Like you can see a trend, you like squint and it's not like a complete blob. You can see a trend, but like there is variation within it. And people are actively investigating how like what that, like what causes that variance, like what environmental factors could change it, like if we could like therapeutically change it, like there's options.
I mean, presumably part of this is like they're looking at like the length in some cell, but like what we were talking about before is kind of like it could be different in different cells, right?
Yeah. Like the protein could be different amount, or it can accumulate differently.
Yeah,
Like
just the number of repeats could accumulate differently, right?
Yeah, the number of repeats, the protein also could be cleared differently in like different people. Like there's so many things we don't know. Like we know so much and yet we know so little. It's is the answer to yes, possibly. But I'm going to show you the last landmark. So I'm going to go back to this timeline. And so in 1996, they had the first mouse model of Huntington's disease. And so I feel like this is the last thing on this timeline because it actually is like the start. This is where people started actually looking carefully about like what causes disease and actually how to alleviate it using not anecdotal, like yeah, patient looks shaky, let's give them some like antipsychotics. Like let's look for a mechanistic way of dealing with this disease, which is exciting. So we are running really, really out of time. So I'm gonna fly through some of these more exciting things. So here are like some explanations for why it's pathogenic. These are all hypotheses for why it's pathogenic, and I have a slide for each. So here is the mechanism. So you have this gene that's being made, and it's like misfolded, and then it makes like these globs, and these globs could basically make like these proteins could like be like kind of like amyloid plaques, kind of like you see in other kinds of dementia, like Alzheimer's, for example. You'll get these plaques. So, for example, here is amyloid-like protein aggregates from Huntington's disease, and you can see that in the brain tissue, and only in the brain tissue of these patients, they'll have these aggregates that like are pleated, and the protein shape is like amyloid plaques. Here you can see that they aggregate similarly to plaques in brain tissue from different kinds of disease. Here is like one of the other explanations for why it's in the striatum is like the downstream transcriptional pathways that Huntington's targets is only in neurons of the striatum. So like you can see here, like here's a bunch of genes. For example, JUNB is like expressed. Oh, sorry, enkephalin is like mostly in the striatum. Like this is in the cortex. JUNB is in the cortex. You can see like these different genes that really are targeted in the striatum. Like these transcriptional profiles are affected in Huntington. So the second column
So
is the
even though that
Huntington's models.
Huntington gene is present in all cells, kind of it only is really affecting the cellular physiology, like it's affecting a subset of other genes, and kind of if you don't need to use those genes, kind of your cell's fine. But if
Yeah,
you do,
correct.
you're in trouble.
Yeah. Yeah, so these are like downstream transcriptional, like that's what like could cause the disease. It's like what's downstream of Huntington's, like what does Huntington do? So these are possible differences. This is another transcription alteration. It's the same concept. Another one is like metabolism, like you can alter the actual metabolism of the cell. And so here it's just like I think this is what is this? This is radioactivity of this. So they basically gave people like a dye, like they like drink like a milkshake, and they look at the amounts of blood flow, like oxygenated blood flow, and like you can see, this is a really horrible graph. I should find the original and make a new screen capture, because it's like basically it has like less oxygenated blood in the striatum, so it's like starving, like the cells are starving to death from like not enough oxygen, basically. That's like one of the ideas. There's like so this is also metabolism, talking about like labeling it. So these are all and then there's this like proteasome. These are all just like different ways hypothesized and like highly correlated reasons that Huntington's this altered version of Huntington gene could affect could cause a disease. And none of this would be possible without like mice model of Huntington's disease, which is really exciting. And let's go into the cool bit, because I wanted to end in a positive note. So because we know a lot of things, but we can study how it can be how the disease comes about. We can also try and alleviate it. And so there's a bunch of there's a bunch of stages where you can affect it. So here is a graphical depiction of like all the places that we can affect it. So here's DNA, here's the chromosome 4, here's the CAG repeats, and one of the things you could do is you could go in and straight up CRISPR, use zinc fingers, talons. You can like CRISPR out those extra bits. That's one possibility. Then once the DNA is on the way to being transcribed into mRNA, you can use these small molecules to like attack it. On one small molecule, I can go over. It's called ASOs. They basically will bind to the mRNAs, and when they expect mRNAs and there's like it basically makes a pseudo DNA. It just gets cleaved. It just gets broken up, because they're like, that's not an RNA anymore. Break it down. And so you can like attack. This is another way you can intervene. And so the whole idea behind these two versions is like make less of the Huntington's disease version, like make less, because like the humans have two copies usually of like one Huntington's, which is unaffected, and then one that is affected
Mhm.
in the most patients, except for those juvenile patients. Um, and so like if you just like shut down the one that's making all the aberrant versions, you can like have the correct amount of Huntington's,
Oh, nice.
like the
Yeah.
correct gene. And so like that's like the idea. So another way is to use RNAis. So this is like inhibitory RNAs. I basically just target the version that's folded that has way too many way too many CAG repeats, and like get the polyglutamine ones, leave the other ones alone, so you can try an RNAi, shut those down. So that's this version right here. And then like as it's being made into protein, you can also target the protein directly, right? You can this is Protax, for example. This will just target the actual protein being made and like degrade it,
Just
like make
reduce
it
it.
into
Yeah.
yeah, just like reduce the ones that are like the wrong length
Your
and
own shape.
size.
Yeah.
Yeah, so
Yeah.
like these are all like ways that are currently being used to target Huntington's and like really like rescue people from this disease. And like many of these are in trials like today, like right now. If you have Huntington's, you can sign up for a trial. and
Oh, well,
um
yeah, I
and
was gonna ask, are they like preclinical or like stage 1 or like where are
Yeah,
they?
they're all in various stages, like stage 1, stage 2. So, during the talk I'm giving in the lecture tomorrow, I'm gonna go into like one of them, which I think is really exciting, which is the ASOs. I learned about it at SFN actually. I didn't know anything about ASOs. I was just like, oh, you're doing RNA things, it must be RNAi. No, I learned about ASOs recently, and I was like, oh, this is a cool, like we should talk about it. Um, this is yeah, this is just me talking about ASOs and how it works. Oh yeah, I even have a thing. So here is the Huntington genes during transcription. When it's an RNA state, it's single stranded, and then basically ASOs are like a single stranded thing that goes in and binds to the Huntington mRNA. And because it's bound, they're like you have like systems in your cells that are like, break that down, it's not supposed to be there, because it's not supposed to be like this weird fragmented
You're not
hybrid
supposed to
DNA
have
thing.
RNA bound to DNA, so.
Correct. And so if this is bound, or like this double stranded thing that's like partially, it's just like hanging out. It's like, that's a problem. Let's break it down. And so it's just using that system. It shows that it can go into neurons and astrocytes and all kinds of neurons. And the only like drawback to this particular way is that you have to so like CRISPR would get it once and then it'll be forever. Because this is an RNA, like it works on the mRNAs, you have to like constantly give this to the person. So you would have to basically give them an intrathecal injection like periodically, so that it keeps targeting it. And so this is just a video showing you how intrathecal injections
And
work.
so intrathecal is into the spine?
And yeah, into the space between the spinal cord, and like it's that space, like where the CSF lives
Right.
in your
And
back.
so
So
the
it's
goal is to get it into the cerebral spinal fluid, which kind of circulates through the brain and the spine.
yeah,
And then that
yeah.
way you can get it into like all the cells that should be affected.
Yeah, and in fact, the way they do this is that they inject you. So it's kind of like what is that procedure that they when you're like giving birth?
Lumbar
Like it's like an
puncture?
epidural. Yeah, it's a
Oh
lumbar
yeah.
puncture.
Oh
So
yeah.
it's like it's very similar to well, I feel like most people would kind of know what an epidural is. It's like that, but instead of taking injecting in like anesthetics, you're injecting in um drugs, and it actually goes straight into your CNS system. So like it is bathing your spinal cord, and because it's like the CSF only moves when your heart when your blood moves, they actually make you walk around. So like they'll inject you and then immediately make you like walk on a treadmill. Or make these patients walk on a treadmill, so that it would like pump your heart rate up, and then that would in turn move the CSF around and like hopefully get it into your different ventricles, and like bathe your brain with these RNA, the ASOs, and that would allow it basically less Huntington's gene. Do I have more slides? I think I have oh yeah, that's just the slide showing that it works in mice and in humans, and it like actually works in like a dose dependent way, which is exciting because it's like if you put a little bit, it works a little, and if you put more, it works even better. Like that's something you want when you're like testing if something works. They actually this worked almost too well because like when they were picking, this went through stage clinical 1 and 2, or no, I think in clinical 2 they halted it because they picked 2 doses, and I think they picked like the 2 highest doses, and it was like way too high, and it basically caused an inflammatory response. And they're like, we need to like make new markers to be like this is too high, and you have to titer it for each person. And so they're going back. The bad news is they halted this particular trial. The good news is that they're actually going back and they're doing it again with like different doses. So there is still hope. And with the last couple of minutes, I want to say like 10 years ago, I took this class that I'm now like teaching, and it was such a depressing class because all we heard of was the first half of the class, which was this is depressing, this is how it's depressing.
Yeah.
Hope you don't get it because it sucks. And that was 2014. Like I took this class in the spring of my senior year in college, and like I had no idea like CRISPR was around the corner, these ASOs are around the corner, and like this disease, like solving this particular disease also means any like if this work any of these work, you could target any other monogenic disease. Do you know what I mean? Like this could work for all kinds of diseases that like some that I mentioned already. Like that's such an exciting and hopeful way. Anyway, I wanted to end in a positive note, which is like there's hope. Like, and science is cool. join join
Yeah,
us.
like that's a really cool that's a really cool just like wow, like we're starting to see progress with this. That's really cool.
Yeah, like so much progress in the past 10 years, like since I graduated college. Like I just remember in college I was like I don't think I can study diseases. I would be so depressed. And I'm so glad
Now
someone
you're
else.
like, oh, we're starting to be able to do stuff. Yeah.
Yeah, we're like, "Oh my gosh, we're solving things. Things are happening." Like, damn, it's a good place. It's a good time to do science. But anyway, that's my lecture for tomorrow in class.
Thanks. Yeah.
Thank you for being our class of 1. Did we have any comments? Oh, was
We
it
have a few
Revive B?
comments, yeah, and some lurkers. Yeah.
Oh, okay.
Yeah.
Cool.
Yep.
So anyway, that was our
Yeah.
Huntington's episode.
Yeah. And we will be back. So I think next week I'm gonna be in Hawaii. So I don't know, is
Let's
Nadir
do
gonna
a
be there?
let's do a
Yeah. Okay.
hiatus because next
Yeah.
week I'm also moving house.
Oh, thanks, everybody.
Oh,
Yeah.
thanks, Ravi. Yeah, we'll do a let's do like a 1 week break and then we'll be back because
Okay.
Nadir
Yeah.
will also
Okay.
be back from Singapore.
Yeah. Yeah. Cool.
Which will be
So next
great.
week we will not be there, but the week after that we'll be back for a show. So yeah.
Thanks for listening.